Hi, I'm really excited to get to present CellExplorer for this year's Neuropixel course. CellExplorer is a framework for analyzing and characterizing single cells, and it was published in Neuron last year. As you know, neurons are morphologically very diverse, and neurophysiological studies makes it clear that this diversity reflects extensive functional diversity. The long-term aim with CellExplorer is to tie the large diversity of neurons to electrophysiological features, what we call cell metrics. Cell metrics are features describing single cells that can be derived from EFIS data and other measures, and we separate the metrics into three levels. The first level metrics are biophysical features of the single cells, so this includes the spike waveform, the physical location, the autocorrelograms, and shown here are two example cells with the average waveform recorded across the channels and the location projected on a multi-shank probe, and the ACG of course. And from these basic metrics, we also do a rough cell type classification as captured in panel D, which shows putative, interneurons, and pyramidal cells. The second level metrics are population dynamics, so how cells respond across population. This includes the monosynaptic connections, spike triggered average to physiological events, response to LFP and oscillations, and to state transitions. And the third level metrics are brain states and overt behavioral correlates. This describes how the cells respond in various brain states and correlates behavioral measures such as body temperature, heart rate, pupil diameter, and whisker movement. We built CellExplorer as a framework around these cell metrics consisting of three components. The first component is a processing module, a streamlined pipeline that calculates the metrics I just mentioned, the temporal metrics, spike waveform metrics, monosynaptic connections, but also spatial metrics if you're doing a behavior, if your subject is doing behavior, and PHDH for manipulations and physiological events. For data inputs, it requires the experimental metadata, the process spike data, and the raw data. The second component is a standardized data structure. This serves as a common language with tailored data containers and structured arrays that are easily interpretable, machine readable, and expandable. In today's demo, we will focus on the session metadata and the cell metrics. The third component is the graphical interface. This allows you to characterize and visualize your cells using the calculated metrics. It's a very responsive and interactive data representation with numerous built-in plots and styles. It offers reference and ground truth data and dimensionality reduction directly in the GUI. And finally allows for manual creation. All this serves to streamline the processing of single cells and offer standard language to use across labs. Each of these components are expandable so you can add your own custom code, custom metrics, and also custom plots. Everything is coded in Matlab. It uses Matlab structure and mat files, but metrics can also be exported JSON and NWB files. And then there's a dedicated GitHub repository and a website called CellExplorer.org with documentation and tutorials to help you get started. So now I would like to show you a demo in Matlab where I will focus on 4 points. How to prepare the experimental metadata, processing the cell metrics with the pipeline, exploring in CellExplorer, and finally use Neuroscope 2 to inspect the raw data, spikes, and the cell metrics. Okay, so here we are inside Matlab, and I have a Neuropixel recording from a pilot study I did with the 384 channels. It's 200 gigabyte, about 2.5 hours. And this data set you can see on the left, it consists of the raw data, metadata from KiloSort NeuroSuite, and then spike sorting data from Phi. And now I'm using a template script to import these metadata from these files, but also filling out the rest of the details as good as possible. And then I'm using a graphical interface to visualize this for manual creation afterwards. And here you see the interface. I'm gonna do this a bit quick in interest of time. So we are recording from a rat, log image rat, from a male. And you can see the metadata for the extracellular data has been filled out, the probe layout, the electrode groups, the channel coordinates also been generated. We can see what we have. This is generated from KiloSort. It's not using all the channels, and we want to make this a little bit better. So we're gonna create a poly 2 layout just for the visualization, like this. We also want to make sure the format is correctly filled out as Phi of the spike sorting, and this is good. So now we're going to run the pipeline. The pipeline generates a number of files, the cell metrics, it saves a session, it saves spikes, and all these files and structure you can learn about on the CellExplorer website. I'm going to pause the video now because it takes a few minutes to run. Okay, so now this processing is finished, and we can expect it with CellExplorer. It took about 4 minutes to complete. So this is CellExplorer for a single session. I can click cells up here. They are basically consisting of points. The top are what we call groom plots. The top left is a custom group plot where you can change the data shown. We have the classical separation plot, which shows the dimension used for the separation of cell types, putative cell types. And then we have a dimensionality reduction plot on the right. On the bottom, we have the single cell features. On the left, we have parameters for the custom plot, filters, settings for the single cell plots. And on the right side, we have navigation. Here we can navigate our cells. We can do classification, creation. So here, let's say I want to change this to wide waveform neuron. I can also do that with the polygon selection. I can select a couple of cells by drawing a polygon and then assign cell types, for example. There's a lot of actions I can do on a group basis. And on the bottom right, we have the metrics of the current active cell. Okay, we're going to save this because I want to show you guys how it looks like in batch mode. So now we're saving the settings back, our manual changes back, and they are actually being saved to the cell metrics file. So you can work manually with your data like this. And this one I'll show you guys now with a batch of session. This is from mice and rats. It's a 3,600 cells from 111 sessions. And now I'm loading the cell metrics that I combined already. And you do this using a cell metrics batch script where you supply the base path and the base names to the data, and then it creates cell metrics across the whole batch that you can work on. So here's a session with the 3,600 cells, and again, it's very responsive. I can click around to any given cell. I want to focus now on the bottom plot. So here you have the waveform on the current cell. We have the location of it across the channels and the different cells recorded across the same session. We have the autocorrelograms with the firing rate across time, but we can also show this across the cells. So now I'm showing you all the way from this session. The narrow waveforms are shown in blue, the pyramidal cells in red, and you can really see how nicely they separate. We can see the ACGs across the session. We can see the firing rate across cells. We can also represent this as an image representation. We can also see the waveforms across the channels. We can see them where they are projected on the cell, now color-coded by the different types. We can see the amplitude that basically goes into the estimation of where the cells are. So this is the current cell in red. These are the other cells from the session. We can also create firing rate maps. So here we have potentially 2 place fields from a place cell. We can see how the cell responds to events, sharp wave ripples. And we can also see where it is according to events. So this is kind of like a custom plot where you're showing the average ripple. We can also show rasters of that cell. So on the bottom left are the rasters of the spikes. Now with the extra dimensionality, the CV2 on the y-axis, we can see the raster for the current cell across the session, highlighted in red. And another plot I really like is the connectivity graph. So here I'm showing you all the connections that determined in this data set, all the monosynaptic connections. And you can see our current pyramidal cells here is projecting to a narrow interneuron. Okay, so let's go to the top plot. In the top left, you can change the data shown. You can change to a 3D plot that can actually be rotated. So you can very easily inspect your data. You can change the scales. Let's say you want to have log scales. You can do this also by filters and changing the group. So now, let's say you only want to look at the parameters of the narrow interneurons. You can very easily do this kind of filters. You combine that with different data groups. Let's say we want to see how this data looks across species. So now we have rats and mice. We can also do this like a very nice rain cloud plot. And again, everything is interactive. You can interact with the data here. So now I'm showing you differences. We can change the data going in. So let's say you want to know the trough to peak variations across species, or maybe just want to know this across cell types, which very nicely captures the difference. Okay, so that's it for CellExplorer. I want to show you guys as well Neuroscope 2, which is a graphical viewer for EFIS data that's really taking advantage of all these data containers we have. So this is raw data from a session. It's bilateral implants where the top channels are from one hemisphere and the others are from the bottom is 2 6-channel probes, and the color code are applied automatically to the probes. And now I'm navigating the data. You can see it's very responsive. I can zoom into any given point. I can even create a small zoom in window where I can do measurements right away. I can do filters. I can filter the trace directly. And again, this is done live as I'm viewing the data. We can do spike detection on the data and find the spikes. We can also go in and actually use the spikes we would detect before. We want to change the color scale here a little bit to be able to see them better. So now I'm making the traces grayscale. Now I can actually also extract the wavefront from the cells we see in this window right here that's being displayed. I can use the cell metric we created before the groups, and I can actually go in and color code both the rasters up here. I can do filters by this type, but I can very easily use all these kind of cell metrics we have to filter and apply here. We can also use event data. So for example, the ripples I mentioned before, we can go in and we can inspect them. We can see how our cells respond to ripples by looking at the fine rate below. We can look at spectrograms. We can do noise across channels. There's a ton of things you can do in here as well. And you can use behavior data as well. So I think this shows you the power of having a standardized format. Last thing I want to say is if you want to learn more, you're welcome to contact me. I'd be happy to give a more in-depth demo if you guys are interested. Thank you.